guys. Okay, so welcome to our second lecture on specific host defense. I just like to point out that this lecture is going to be much shorter than your normal lectures. Without review questions, it's kind of like 15, 16 slides, something like that. I made it this short because I just want to give you guys a reminder here. Please, those of you who are doing the lab extra credit, for the people who attended the in-person labs, please be sure to finish those up. I made this lecture shorter with that intent that you guys, have some extra time, talk to your group members, get those hard copies to Kieran. The due date is pretty soon, so please make sure you guys, the ones of you who are interested in doing so, please make sure you do that and then complete that assignment. Okay, so specific host defense part 2. So again, just as a bit of a review here from last time. Remember last time we spoke specifically about this left half, so the B cell half of the diagram, but both B cells and T cells originate from stem cells in adult red bone marrow or in the fetal liver. As some cells then pass through the thymus, which will then emerge as T cells, while others will go through the bone marrow and become B cells. Both types of cells then migrate to lymphoid tissues such as the lymph nodes or spleens. So again, last week we chatted about this side. Today we're just going to talk about this. We're going to shift focus and talk more about the cellular immunity. So these are another breakdown of the humoral and the cellular immune systems. So because we went through the just a bit of an overview here, remember that you will have a B cell will bind to an antigen for which it is specific. Remember the specificity we discussed. A T-depressant B cell will then require the cooperation of a T cell or T helper cell. We'll discuss more about them today. The B cell, often with the stimulation of cytokines from the T cell, will then differentiate into a plasma cell, and some will become memory cells. You can see them differentiating out to memory cells. There's your plasma cell. Your plasma cell will then proliferate and produce antibodies against the antigen. So there's that happening. So moving on to the other side. So intracellular antigens are expressed on the surface of an ABC, which is essentially a cell infected by a virus or a bacterium or a parasite. So now what happens is a T cell will bind to a MHC. We'll talk more about them later, so I'm not going to go, don't freak out about it too much. We'll bind to the MHC antigen complex on the surface of the infected cells, which will activate the T cells with the cytokine receptors. And then the activation of macrophages, which is essentially just enhancing phagocytic activity. And then CTLP becomes an activated cytotoxic lymphocyte, able to induce apoptosis of the target cell. This is the T cell site, just a brief overview. Don't freak out too much. We will be going into more detail. So some important takeaways that I just want you guys to know from this diagram. So I want you guys to, at this point, know that the adaptive immune system is divided into 2 parts. Each is responsible for dealing with pathogens in a different ways. These 2 systems function independently to keep the body free of pathogens. The humoral immunity, also called antibody-mediated immunity, is directed at freely circulating pathogens and dependent on B cells, whereas cellular immunity or cell-mediated immunity is dependent on T cells, which you can see over here. And they depend on these T cells to eliminate intracellular pathogens, reject foreign tissue recognized as non-self, and destroy tumor cells. Okay, so most pathogens that require cellular immune system to fight against is essentially they first need to enter our cells, and they typically do this by entering either the gastrointestinal tract or the lungs, where they encounter a barrier of epithelial cells. Normally, they can pass this barrier in the gastrointestinal tract only by way of a scattered array of gateway cells called microfold or M cells. Here you can see an example of that. This is an M cell. M cells are located within Peyer's patches. This is also, you can see an M cell on a Peyer's patch, which are located on the intestinal wall, and their function is to transport antigens encountered in the digestive tract to contact lymphocytes and antigen-presenting cells of the immune system. So again, they transfer antigens to lymphocytes and antigen-presenting cells. You can see another breakdown of what this M cell would actually look like if you had if you sliced through it. The other cells that we need to be aware of here is your antigen-presenting cells. So here you have dendritic cells. These cells engulf and degrade microbes and display them to T cells. They are found on the skin, genital tract, lymph nodes, spleen, thymus, and blood. And here, importantly now, the recognition of antigens by a T cell requires that they first be processed by a specialized antigen-presenting cells, or APCs. This resembles the situation previously discussed in humoral immunity, in which the B cell served as the APC. After processing, an antigenic fragment is presented on the APC surface together with a molecule of the MHC. APCs then include activated macrophages and, most importantly, dendritic cells. Also, what I want you guys to know about these dendritic cells are, again, just the most important type of antigen-presenting cells, and their structure makes them look like they have dendrites, or have the dendrites of nerve cells, and that's where they get the name from. Another cell that you have to be aware of here is macrophages. These are activated by cytokines or the ingestion of antigenic material, and they migrate to the lymph tissue presenting antigen to the T cells. So what are these T cells? Essentially, T cells act against antigens within host cells and larger microbes, again, intracellular, and they develop from stem cells and bone marrow, as displayed by that diagram we went through, and they are influenced by the thymus gland, where they mature into T cells, followed by migration into lymphoid tissues. There are different types of T cells, which all have different functions, which we'll briefly be looking at now. First of all, we have cytotoxic or TC cells, and as their name suggests, they destroy target cells on contact. You have helper cells. These are Th1s. They activate your cytotoxic cells or other helper cells, macrophages, and some B cells. Remember in that diagram, we have that diagonal line where your B cells required some helper cells in order to release cytokines. This is their function. Then you also have Th2s. They protect against infectious agents and contribute to allergic reactions. And then you also have regulatory cells, so TRs. They regulate immune response and help maintain tolerance. Important here, these cells also kill T cells that attack host tissues. So they also prevent autoimmune diseases in that example. So how does antigen recognition in B cells and T cells occur? It's very similar. That's why we have them, why we're just going to discuss both B and T cells. So T cells are similar in many ways to immunoglobulin molecules. Both have variable regions. Here you can see the variable regions of both. Very rudimentary diagram, but your textbooks unfortunately do not have a good example of this. And then these antigen binding sites can recognize a wide variety of antigens. However, just like in antibodies, each T cell receptor is unique. Some other similarities that you should be aware of here: both are made up of pairs of subunits. These are alpha, beta, gamma, and so forth, which are themselves members of immunoglobulin superfamily, and both recognize a wide variety of antigens via an N-terminal variable region. Again, that's that portion. And these antigen recognition subunits are associated with invariant chains of the T cell receptor, the CD3 complex, which form a critical signaling function. Okay. So now we will be looking at cell-mediated cell cytotoxicity. So I think it'll be best to just first give you some background here in that T cytotoxic cells, despite their name, are not capable of attacking any target cell as they emerge from the thymus. Rather, they are precursors to cytotoxic T lymphocytes, CTLs, which have this capability. This differentiation requires a sequential and complex activation of the precursor TC by an antigen processed by a dendritic cell and integration with a TH cell, so a helper cell, and co-stimulatory signals. The resulting CTL is an effector cell that has the ability to recognize and kill target cells that are considered non-self. So essentially here, that's a very complex way of looking, but I think the pictures might make this a lot easier. So in the first diagram, we have a virus-infected cell. So this is an example of an endogenous antigen. Here we have specifically MHC class 1. Again, just as a reminder, MHC is your major histocompatibility complex, and there are two classes of them. There's class 1, which is used in this case. So this would, and they are exclusively present on CD8 cells, where class 2 is exclusive to CD4 cells. So anyway, and here you see the processed antigens, the MHC class 1 being produced, and now you will find the processed antigen is presented with the MHC class 1. So see how this antigen is now present in this MHC class 1, in the major histocompatibility complex, which will then essentially have it recognized by your helper cell. So important here, normally under these circumstances, a normal cell will not trigger a response by a cytotoxic T lymphocyte cell, but a virus-infected cell here or a cancer cell will produce abnormal endogenous antigens. That's these molecules over here. So without these molecules, the MHC1 will never have this form, so it wouldn't be recognized by the T helper cells. So, but once that is prevalent or present, the abnormal antigen is presented on the cell surface, as is illustrated there, in association with the MHC class 1 molecule. And this will bind the T1 helper cell, which will promote the secretion of cytokines. Here you can see the cytokines being secreted. The cytokines will activate a precursor CTL. Again, this is that cytotoxic T lymphocyte, which will produce a clone of the cytotoxic T lymphocytes. And then this CTL will then induce the destruction of the virus-infected cell by apoptosis. Important here that it uses apoptosis. It doesn't simply lyse the cell. We'll talk a bit more about that later on. But afterwards, the cell will be lysed after apoptosis. So apoptosis is just cell death. But here again, the cell will be lysed at the end. Okay. So now we're going to be looking at some helper cells. So to activate a CD4 helper cell, at least two signals are required. The first signal, so hang on, let me just, this might help. So again, now we're going to be jumping to this cell over here. How is this cell activated? Okay, anyway, so just to recap here, just to restate. To activate this cell, we need two signals. The first is the signal that binds the TCR receptor to the processed antigen, and the second signal requires a co-stimulating cytokine such as IL-2. There are some other examples too, but it requires one of them. Once activated, the T helper cell will secrete cytokines that affect the effector functions of multiple cell types of the immune system. So here we have an APC, which will encounter and ingest a microorganism. The antigen is enzymatically processed into short peptides, which you can see over here. There are short peptides. which combine with the MHC class 1. Remember, this is our major histocompatibility complex, and will then be displayed on the surface of the APC. A receptor TCR, again, just here, just for clarification, APC is a dendritic cell, just so that you are aware that there's our definition. A receptor TCR on the surface of the CD4 T helper cell, or the TH cell, will then bind to this MHC antigen complex. That we saw that in the previous slide, and here we see it just more in a close zoomed in view, which will induce a toll-like receptor. The TH cell, or APC, is stimulated to secrete a co-stimulatory molecule. And then these two signals are what activate the T helper cell to produce cytokines. So here you can see cytokines are again produced. And here's our co-stimulatory molecule, which is needed for this molecule to recognize, okay, I should now start producing cytokines. The cytokines will then cause the helper cell to become activated, essentially. So it will recognize a dendritic cell that is producing the co-stimulatory molecule, and then it'll become activated. Okay. So moving on to helper T cells. So this diagram that we have here depicts a lineage of effector T helper cell classes and the pathogens that they target. So once a T helper cell is produced, the proliferating TH cells will differentiate into population of substrates such as TH1, which you can actually, this is sort of an wrong version. So here we have TH1, TH2 at the bottom, and then TH17. They will also form a population of long-lived memory cells, again, so that our secondary response is more efficient than our initial response. The effector functions of these substrates are based on the cytokines produced by the T helper cells, which act on different cells in the body's defensive systems. So now we're going to be discussing these in order. So TH17 cells, what are they? These are situated in the skin and in the lining of the gastrointestinal tract, where they are well positioned to deal with potentially invading microbes. When external pathogenic invaders are detected, TH17 cells stimulate the innate immune system. For example, they can activate cells that synthesize cytokines such as TNF that are responsible for inflammatory responses. TH17 cells also recruit neutrophils to help clear away extracellular pathogens. And then moving on to T2 cells. TH2 cells produce cytokines that are associated primarily with the production of antibodies, especially IgE, that are important in allergic reactions. Remember, E is for the immunoglobulins present for allergic reactions. They're also important in the activation of eosinophils that defend against infections by extracellular parasites such as helminths. And then finally, on this side of the diagram, on the right-hand side, we have cytokines produced by TH1 cells, which activate mostly, related to the important elements of cellular immunity, such as delayed type hypersensitivity, and are also responsible for the activation of macrophages. They stimulate the production of antibodies that promote phagocytosis and are especially effective in enhancing the activity of complement, such as opsonation and inflammation. Opsonation, very important. We will be chatting about this here in a bit. Note that the generation of cytotoxic T lymphocytes also requires the action by the TH1 cell. So the process that we discussed in the preceding slides, they require these TH1 cells. Okay, cytokines and lymphokines produced by T cells. So these are essentially chemical messengers for immune cells. There are a few examples here. You have your interleukins, which aids in communication between different leukocytes, leukocytes being white blood cells. You have chemokines, they induce migration of leukocytes to an area of infection or damage. You have interferons, they interfere with viral infections. Tumor necrosis factor causes inflammatory reactions in autoimmune diseases. And then you have granuloleukocyte, Granuloleucocyte macrophage colony stimulating factor, which is a class of compounds that stimulates blood cell differentiation. Moving on to NK or natural killer cells. This is a type of cytotoxic lymphocyte that is involved in the innate immune system. These are pretty broad because they can attack parasites, virus-infected cells, and tumor cells. These natural killer cells will release perforin when they are close to the target cells, which will form pores in the cell membrane of the target, inducing apoptosis. Apoptosis here being cell death. Important though here, remember when I said earlier, it's important to distinguish between apoptosis and lysis, especially when you look at immunology, because if you lyse a virus-infected cell, you would simply just release the virions. Whereas with apoptosis, it would lead to the destruction of the virus inside. So you need the apoptosis to actually destroy the virus before the cell can safely be lysed. Where if you were just to lyse the cell from the get-go, you would just be releasing the virus everywhere and sort of compound the problem. So this kind of just reviews that, where a viral infected cell lysis leads to the release of virions, whereas apoptosis leads to the destruction of virus inside the target cell. Very important distinction in regards to immunology. Okay, again, here we just have a quick overview of the two processes that we have discussed in our last two lectures. I'm not going to run through all of it again. I just want you to remember the primary differences, or just look at this and remember the primary differences. First, there's humoral immunity, also called antibody-mediated immunity, which is specifically directed at circulating pathogens, freely circulating pathogens, and depends on the B cells. So here you have your B cells. And then you have cellular immunity on the other side, also called cell-mediated immunity, which depend on T cells. To eliminate intracellular pathogens and reject foreign tissue recognized as non-self, and also destroy tumor cells. So here's just a little video for you to review our work, and then another one that's really helpful. This YouTube video, the guy actually draws out the entire process. He provides a bit more detail. I won't test you on anything in this video, but it is really, really good. Just to improve your own understanding and just cover it in more detail. I'm going to do quick 4 review questions, and then let you guys get cracking on those lab reports if you are not done with those yet. So review question 1, true or false? Cell-mediated immunity depends on T cells to eliminate intracellular pathogens. The answer, of course, here is true. Microfold cells transfer antigens to lymphocytes and antigen-presenting cells. APCs. Is this true or is this false? Okay, moving on to the answer. It is also true. And I'm just going to ask you some open-ended questions. Where are dendritic cells found? I'll give you guys a few minutes or few seconds here to think about that. Please pause the video if you want to test yourself. They are found in the skin, the genital tract, the lymph nodes, spleen, thymus, and blood. And then moving on to a review question number 4. How many signals are required to activate a CD4 plus helper cell? Think back. We discussed this earlier. Okay, and I'm going to move on to the answer here. So the answer is that you need 2 signals. And if I ask a question like that, this would be sufficient for a 1-mark answer. But if, this was maybe a 3-4 mark or a longer form question, I would expect more detail. I would also structure the question differently, of course. But just as a reminder for you guys here, also, the first signal is the binding of the TCR, so the receptor, to the processed antigen. And the second signal is requires a co-stimulating cytokine. Remember those little white molecules on our diagram. Okay, guys, that'll do it for today. I hope you can use the extra time to finish those lab reports, and then I will see you in the next lecture. Okay, cheers.